Uh, I'm really sorry. Good afternoon to all of you. My voice is audible. Please let me know. Sorry for the disturbance which we had before. Please let me know. Please let me know everything is fine now. All of you have to reply me only once so that I will start. Okay. So are you getting me or not? Let me know. Are you getting me? Hello? Okay, okay. Thank you. Okay, okay. Thank you, Archana. Thank you, Diksha, Rajneesh. So, okay, shall we start now? Shall we start now? Because we are just taking. Shall we start now? You have any query regarding the review paper or something? You have to let me know. Please let me know if you have any query regarding the review paper. Because it is 8th of December. You have to submit by 10th of December. If you have any query or something, please let me know. Sandra, what is your question? Basically, I am not getting it properly. No, there is. Just answer me. I am not getting your point, Sandra. Please elaborate it, please. Yes, 10th of December, you have to send me the topic and the abstract along. So yes, you have to send me the review paper. You have to send me the title or the topic of the review paper along with the abstract. And what is your question basically, Sandra? There is any format? No, there is not only any format. You have to type it in the MS Word. It includes your topic as well as the whole information which you want to tell us in the review paper. Getting my point? Yes, you have to write in abstract, you have to write the thing which you want to elaborate in the whole review paper. The important points, the highlights which should be there in the review paper, you have to add only those lines in the abstract only. Because abstract is the main one, the person will read the abstract and then they will able to know what is the main content in the review paper. Getting my point? So your abstract should be the nutshell of your whole review paper. Or basically what you want to say in the review paper, that is there in the abstract only. Is there any another question regarding the review paper? I am just waiting the students to join, otherwise I will start the session. We are already very late today. Do you have any another question also? So do you have any question regarding the review paper, or shall I start the shall I start the lecture now? So today we have to cover the enzyme kinetics part. We have done little bit about the enzymes yesterday also. So today we have to done some another topics which are there in the enzyme. So we will try to complete it today. So today we will try to complete the enzymes today. Enzyme regulation, isoenzymes, ab enzymes, basics of enzyme immobilization. So I will try to complete the part of enzymes. So are you getting me? My voice is audible? Please let me know so that I will be able to share the screen. Let me open the screen. Are you getting me? My voice is audible to all of you. So this is the enzyme which we have to start. Just let me know. Are you able to get it? The screen will be okay soon. Just give me some time. Sometimes it happens because of the network issues only. Otherwise, the screen is visible to all of you. So, is it visible to all of you? The screen is visible. So, these things we have already done yesterday. Types of enzymes, the nomenclature which is there in the enzyme, classification of enzymes, the properties of enzymes. So, today we will try to start. Yes, we have to start from this only. So is it visible to all of you? Now Diksha, is it visible? Diksha, please let me know, is it visible? Please let me know, is it visible to you now? Any another one? Any another? Archana, the screen is visible to you? Archana, Rajneesh, Sandra, the screen is still blurred. Is it visible to you? Let me know. Are you not getting the screen? This screen is not visible to you. Let me know only so that I start the session. Is it visible to you now? Okay, okay, okay. Because my side the signal is okay, I am able to get the screen very right. So maybe because of network issue, you are not getting it properly, Diksha. Because the screen is okay here. Okay. So shall we start now? What is the homework which I gave you yesterday? What is the homework which I gave you yesterday? Please let me know what is the thing which I asked you to complete yesterday. Anamika, Rajneesh, Diksha, have you done the homework? Any of you? Please let me know. Have you done the homework? Yeah, different classes of enzymes. Very good. Then another question is, please answer. What is the question? The homework which I gave you yesterday. I gave you how many questions yesterday? Yeah, ribozymes and induced fit theory also. Please write the questions and let me know. Have you done it or not? Please ask. Please let me know the questions which are there yesterday. Different classes of enzymes, ribozymes, and one more question is there. Which is the that question? Let me know. Okay, good, Archana, good. Yes, LDL and HDL. Please let me know. Thank you, sir. Thank you so much for giving this opportunity to all of us. Thank you, sir. Thank you, sir. Bye. Thank you, sir. Hello. So shall I start now? I have to complete the enzymes today. So I am just going to start the presentation. Hope my voice is audible. My voice is audible to all of you. Please let me know. My voice is audible to all of you. Please let me know only one. So that I will start the enzymes part. Rajneesh, my voice is audible? Hello. Okay. So I'm just going to start with the remaining part which is there in the enzymes. So yesterday we have started enzymes, and in the enzymes we have discussed all the important points, the characters of particular enzymes and everything. So today I will going to discuss further about enzymes. So in it we have to start today the Michaelis-Menten equation, or you can say Michaelis-Menten hypothesis. So it was in 1913 sorry. It was in 1913 while studying the hydrolysis of sucrose catalyzed by the enzyme invertase proposed this theory. Their theory is however based on the following assumptions. The assumptions are only a single substrate and single product are involved. The process proceeds essentially to completion. The concentration of the substrate is much greater than that of the enzyme in the system. An intermediate enzyme substrate complex is formed. So, intermediate enzyme substrate complex is formed. The rate of decomposition of the substrate is proportional to the concentration of the enzyme substrate complex. So, the rate of decomposition of the substrate is directly proportional to, or you can say it is proportional to, the concentration of the enzyme substrate complex. So this is basically the Michaelis-Menten equation, or you can say this a hypothesis. This theory postulates that enzyme forms a weak bonding complex with the substrate. This enzyme substrate complex on hydrolysis decomposes to yield the reaction product and the free enzyme. So the enzyme substrate complex on hydrolysis decomposes to yield the reaction product and the free enzymes. Okay. So this is the equation which is given by Michaelis and Menten. Like from enzyme plus substrate we are getting enzyme plus substrate complex, and at the end we are getting enzyme plus product. This is the equation basically which is given by the Michaelis and Menten, and this is the graph of this Michaelis-Menten hypothesis. So according to them, that V is half by Vm, Km is numerically equal to the substrate concentration, or in other words, Km is equal to the concentration of the substrate, which gives half the numerical maximal velocity. So it is noteworthy that for any enzyme substrate system, Km has a characteristic value which is independent of the enzyme concentration. So this is the significance of this Km and Vmax value. The Km value is used as a measure of enzyme's affinity for its substrate. The lower the Km value, the higher the enzyme's affinity for the substrate and vice versa. Km value also provides an idea of the strength of binding of the substrate to the enzyme molecule. The lower the Km value, the more tightly bound the substrate is to the enzyme for the reaction to be catalyzed and vice versa. So Km value indicates the lowest concentration of the substrate the enzymes can recognize before reaction catalysis can occur. So an enzyme's Km describes the substrate concentration at which half the enzyme's active sites are occupied by the substrate. So Km value is also used as a measure of the substrate concentration when the reaction rate half maximal, that is Km is at half of the V maximum. The maximal rate reveals the turnover number of an enzyme, that is the number of substrate molecules being catalyzed per second. This varies considerably from 10 in the case of lysozyme to 6 lakh in the case of carbonic anhydrase. Getting it? So this is a Michaelis-Menten equation when we are getting the enzyme substrate and later we are founding the enzyme plus product. So you have to keep this equation in mind only. It is the equation which describes how reaction velocities varies with the substrate concentration. So the velocity varies with the amount of substrate. How much amount of substrate is there, according to which the velocity will vary. And this is the initial velocity, Vmax is the maximum velocity, Km is the Michaelis-Menten constant, S is the substrate. So velocity is dependent upon the substrate concentration. Getting it? So these are some of the assumptions of the Michaelis and Menten equation. So the assumptions are relatively concentration of enzyme and substrate, steady state assumptions, and initial velocity. These are the some of the assumptions according to which we are getting the graph. This is substrate concentration. Now this is the pharmaceutical importances of all these enzymes. So enzymes are basically they are involved in all physiological process which makes them target of choice for the drugs that cure human diseases. So applied enzyme kinetics represents the principal tool by which applied enzyme kinetics represents the principal tool by which scientists identify and characterize therapeutic agents that selectively inhibit the rate of specific enzyme catalyzed processes. So enzyme kinetics thus plays a role in drug discovery as well as elaborating the mode of action of drugs. Now we have another term which is known as inhibition. What is basically inhibition? It is the prevention of an enzyme process as a result of interaction of inhibitors with the enzyme. So inhibitors are the any substances that can diminish the velocity of an enzyme catalyzed reaction. So basically inhibitors will stop the reaction. So they will diminish the velocity of an enzyme. Catalyzed reaction is called as inhibitor. So they are just stopping the rate of reaction. So these are the types of inhibition. We have reversible inhibition, irreversible inhibition, and in the reversible we have competitive, uncompetitive, mixed and non-competitive inhibition. So inhibition is the prevention of an enzyme process as a result of interaction of inhibitors with the enzyme. So inhibitors are any substance that can diminish the velocity of an enzyme. Catalyzed reaction is called the inhibitor. So inhibitors are any substance that can diminish the velocity of an enzyme. Catalyzed reaction is called inhibitor. So these are the inhibition. We have competitive inhibition, uncompetitive, mixed inhibition as well as non-competitive inhibition. Now what happens is reversible inhibition. It is an inhibition of an enzyme activity in which the inhibiting molecule entirely can associate and dissociate from the protein binding site. So reversible is basically four types. We have competitive, uncompetitive, mixed and non-competitive. Now what happens in reversible inhibition? Number one is competitive inhibition. So in this type of inhibition, the inhibitors compete with the substrate for the active site. Formation of ES complex is reduced while a new EI complex is formed. So instead of ES complex, which is enzyme substrate complex, we are getting the EI complex. So instead of substrate, inhibitor will bind. So in this type of inhibition, the inhibitors compete with the substrate for the active site. Formation of ES complex is reduced while a new EI complex is formed. So we are getting the enzyme plus inhibitor complex. So in this type of inhibition, the inhibitors compete with the active site. So the substrate is here and the substrate is not allowed to bind on the active site. Rather in that case, inhibitor will bind. So the example is statin drug as example of competitive inhibition. So statin drugs such as Lipitor compete with HMG carboxy CoA coenzyme A substrate. So basically statin drug inhibitors combine or you can say compete with HMG coenzyme A and the inhibitor site of HMGA coenzyme A reductase that will bring about the catalysis of cholesterol. So this HMG coenzyme A reductase is needed for the cholesterol synthesis. So this is an example of statin. This is an example of competitive inhibition And the example is statin drugs. Now what happen in uncompetitive inhibition? So the substrate will bind to the active site, but we have another site which is known as allosteric site for the inhibitor molecule. Getting my point? So in this type of inhibition, the inhibitor does not compete with the substrate for the active site of an enzyme. Instead, it binds to another site known as allosteric site. So in this, basically what happens, inhibitor does not compete with the substrate for the active site, rather it binds to another site known as allosteric site. Now the examples of uncompetitive inhibition are drugs to treat cases of poisoning by methanol or ethylene glycol act as uncompetitive inhibitors. Tetramethylene sulfoxate and 3-butylthioline 1-oxidase are uncompetitive inhibitors of liver alcohol dehydrogenase. Now what is irreversible inhibition? So in this type of inhibition, covalent attachment of the inhibitor to the enzyme, so this reaction is irreversible. The catalytic activity of the enzyme is completely lost. It can only be restored by synthetic molecules. So aspirin is an example of irreversible inhibition. Aspirin, which targets and covalently modifies a key enzyme involved in the inflammation, is an irreversible inhibitor, or you can say suicide inhibition. It is an unusual type of irreversible inhibition where the enzyme converts the inhibitor into a reactive form in its active site. This is suicide inhibition. Then we have, again, this is uncompetitive inhibition. We already discussed it. You have to keep in mind the example of this, like tetramethylene sulfoxate, 3-butylthioline 1-oxide. So you have to keep the example of these in mind. Now we have another term which is known as activation. What happens in activation? So activation is defined as the conversion of inactive form of an enzyme to active form. So activation is basically conversion of inactive form of an enzyme to active form, which processes the metabolic activity. So the types of activation are activation by cofactors, conversion of an enzyme precursor. So activation by cofactors. So many enzymes are activated by the cofactors. The example is DNA polymerase, horse liver dehydrogenase. So DNA polymerase is a hollow enzyme that catalyzes the polymerization of deoxyribonucleotide into a DNA strand. It uses magnesium ion for the catalytic activity. Horse liver dehydrogenase uses zinc ion for its activity. So many enzymes need the cofactors for its activity. Like DNA enzyme needs magnesium ion, DNA polymerase. Similarly, horse liver dehydrogenase use zinc ion for its activity. So horse liver dehydrogenase uses the zinc ion. Then conversion of an enzyme precursor, passive proteolysis, is a common method of activating the enzyme and other proteins in the biological system. The example is the generation of trypsin from trypsinogen leads to the activation of another enzyme, which are known as zymogens. So this is how one enzyme is able to activate the another enzyme. So trypsin is able to activate chymotrypsin from chymotrypsinogen. So one enzyme is activating the another enzyme. Enzyme specificity is basically what, when the enzymes are highly specific in nature, interacting with one or few substrates and catalyzing only one type of chemical reaction. Substrate specificity is due to complete fitting of active site and substrate. Oxidoreductase do not catalyze hydrolase reaction, and hydrolase do not catalyze reaction by involving oxidation and reduction. So this is the type of specificity: bond specificity, group specificity, absolute specificity, optical specificity, and dual specificity. We already discussed it yesterday also. So this is again different types of specificity which are there in the enzyme. Then the enzyme regulation by allosteric regulation, by using compartmentalization, by enzyme degradation. So what is allosteric regulation? additional sites other than active sites. They are known as allosteric enzymes. So most of the allosteric enzymes are oligomeric in nature, non-reversible binding of effector molecules at the allosteric sites, conformational change in the active site of the enzyme. So these allosteric sites or these allosteric enzymes, they are responsible for the change in the active site of an enzyme. So some enzymes remain inactive. It gets activated at the site of action by the breakdown of one or more peptide bonds. The example is chymotrypsin, pepsinogen and plasminogen. Certain enzymes keep interconverting from active to inactive and vice versa, depending upon the need of the body. Now what is compartmentation? The enzyme remains confined to a particular area of the cell which makes it exclusive. For instance, fatty acid synthesis takes place in the cytosol, whereas fatty acid oxidation takes place in the mitochondria. So synthesis is where in the cytoplasm and the working is where it is in the mitochondria. So control of enzyme synthesis. Most of the enzymes, particularly the rate limiting ones, are present in very low concentration. Based on the amount of enzyme present in the body, enzymes are constitutive enzymes as well as adaptive enzymes. I will share the PPT so that you will be able to understand what I want to say. So we have different types, like we have isoenzymes. What happens here? When the same reaction is catalyzed by two or more different molecular forms of an enzyme, then it is called isoenzyme. Okay. After it, it may occur in the same species, in the tissue or even in the same cell. So different forms of enzymes generally differ in kinetic or regulatory properties. The example is hexokinase 4, lactate dehydrogenase, keratin phosphate, keratin kinase, alkaline phosphate 6, alcohol dehydrogenase 2. Then diagnostic importance of enzymes. So estimation of enzyme activities in biological fluids is of great clinical importance. The enzymes can be divided into two groups, plasma specific, which are known as plasma functional enzymes, or non-plasma specific or plasma non-functional enzymes. So plasma specific enzymes, they are present in the plasma normally and have specific function. Their value is higher in the plasma than in the tissue. They are mainly synthesized in the liver and enter the circulation. The example is lipoprotein lipase, plasmin, thrombin, choline esterase and ceruloplasmin. Impairment of liver function or genetic disorders leads to leads to enzyme deficiency, which is known as Wilson's disease, which is a deficiency of ceruloplasmin. Then we have non-plasma specific. These enzymes are present in the low level These enzymes are present in the low level in the plasma compared to the tissue. Estimation of activity of these enzymes serves for the diagnosis and prognosis of several diseases. So these are known as markers of the diseases. The raised enzyme level may indicate cellular damage, increased rate of cell turnover, proliferation of the cells, increased synthesis of the enzymes. So these are some important enzymes which are there. We have amylase, we have serum glutamate oxaloacetate, alkaline phosphatase, acid phosphatase, lactate dehydrogenase, keratin phosphokinase, aldolase. So these all enzymes, they have specific functions. Like amylase is basically for acute pancreatitis. Serum glutamate pyruvate transferase is for the liver disease, which is hepatitis. Similarly, serum glutamate oxaloacetate transaminase is for the heart attack. So they are quite important for diagnosing all these diseases. Aldolase is for muscular dystrophy. Lactate dehydrogenase, which is LDH, it is basically for the diagnosis of heart attack and liver diseases. So amylase is the number one amylase. So amylase activity increase in acute pancreatitis. It is quite useful in urine analysis, also in the serum analysis. Like serum glutamate pyruvate transferase is quite helpful in diagnosing acute hepatitis, jaundice, and cirrhosis of liver. Alkaline phosphatase is responsible for the diagnosis of brackets, hyperparathyroidism, carcinoma of bone, obstructive jaundice, Paget's disease. Acid phosphate is for the diagnosis of cancer in the prostate gland and in pelvic field. So this phosphatase, acid phosphatase, is quite useful for the diagnosis of tumor marker. Getting it? Then we have lactate dehydrogenase. Now we have three different forms of these enzymes: LDH1, LDH2, LDH3, 4, and 5. They are also helpful in the diagnosis of heart damage during the myocardial infarction. Similarly, keratin, phosphokinase, aldolase, and these are some references. If you want to read enzymes in detail, you can prescribe those books also. Okay. I think that is all in the digestive, in this enzymatic part. Are you able to get it or not? Please let me know. Are you getting it? Are you able to understand it? I will share the PPT so that all of you will be able to understand it. So tomorrow we will start. So this enzyme part is covered right now. So tomorrow we will start with another part which is there, which is lipids, the classification, structure, and function of lipids. Let me Yeah, so we have to start the lipids. Students are very few today, otherwise I want to start it today. I want to start lipids, and then after it we have fatty acids, phospholipids, and lipoproteins. Then we have amino acids and proteins. So I am just waiting for the students, but only few students are here today. So are you getting it or not? Please let me know whether you are getting it or not. So shall we start further or it is okay for today? Is it okay for today or shall we continue? Let me know please. I want to start the liquid. Shall we start it or not? Shall we start it further or is it okay for today? So I think that's all for today. You are not replying to me. Please let me know whether you are getting my voice or not. I am just asking you, shall we start the lipids today or we have to do it tomorrow? Hello, my voice is audible? My voice is audible? Hello, my voice is audible? So I think that's all for today. We'll meet tomorrow because only 5 students are here right now. If you want, I can start the lipids also, but I am just wanting to know whether you are getting it or not. Okay, so shall I continue? Because if we will able to continue it, then we will able to complete it as possible. Because I know some of you have your exams from 15th of December maybe. So I just want to complete the whole thing by 15th of December so that you all will be free. Getting my point? So shall I start now or not? Please answer me if possible. I want to complete the session by 15th of December so that you all will be able to take your exams as soon as possible. Shall I start now? Please reply. Where is Diksha? Diksha, you are not replying me. You are first replying when screen is blur blur. Now I am asking continuously. You are not even bothered to reply me. I request all of you to reply if you are able to listen me. Okay, thank you, Kalpesh. Thanks a lot. Okay, so I'm just going to start with lipids now. So this is you are in the syllabus. We have to do lipids, the classification, structure, and functions of lipids. Yeah. So basically, what are the lipids? What are lipids basically? So lipids are any member of a large and a diverse group of oils, fats, and fat-like substances that occur in a living organism and that characteristically are soluble in organic solvents but only sparingly soluble in aqueous solvents. So basically what are the lipids? They are a member of a large and diverse group of oils, fats and fat-like substances that occur in the living organisms and that characteristically are soluble in organic solvents but only soluble in the but only sparingly, or you can say they are insoluble in the aqueous solvents. So lipids are not polymers but mostly small molecules. They are the chief cellular storage form of energy. They are helpful in the cellular structure and biochemical functions as well. So what are the lipids? They are the heterogeneous class of naturally occurring organic substances. They are heterogeneous class of naturally occurring organic substances. They have a distinguished functional group, or you can say structural features. They are insoluble in water and highly soluble in one or more form of the solvents, like they are soluble in either chloroform, benzene and acetone, but they are insoluble in water. So they are the class of naturally occurring organic substances. They have a functional group as well as structural features. So they are insoluble in water but they are highly soluble in one or more of the solvents like either chloroform, benzene, acetone. So this property sets them apart from proteins, carbohydrates, nucleic acids and other biomolecules. They are widely distributed in the biological world. They play a wide variety of roles in plants and animal tissues. So what are the functions of lipids? Lipids are concentrated source of energy. 1 gram of fat gives 9 calories. It serves as a cushion for the vital organs and protects them from external shock. So lipids, or you can say fat molecules, they are quite important in our body. They serve as a cushion for the vital organs and protects them from the external shocks or injuries. So lipids are the structural material of cells and membranes. They serve as an insulator for our body. Lipids are the carrier or reservoir of fat-soluble vitamins. So in food preparation, lipids serve as a binding agent. It also enhances the palatability of the food. So these are the types of lipids which we have to study in detail. So we have storage lipids as well as we have membrane lipids. So we have two types of lipids. We have storage lipids which are also known as triglycerides, and we have membrane lipids which are known as phospholipid and glycolipid. So now what happens in the storage lipid or in the triglyceride? We have a glycerol molecule, and along with this glycerol we have three fatty acid side chains. So glycerol, also known as glycerin or glycerine, is a simple polyol compound. It is colorless, odorless, viscous liquid that is sweet tasting and non-toxic. The glycerol backbone is found in all lipids and is known as triglyceride. So because we have three fatty acid chains, it is known as triglyceride. Glycerol is an alcohol with 3 carbon, 5 hydrogen and 3 hydroxyl groups. So we have fatty acid chains which are attached with the glycerol molecule. So that is why they are known as triacyl glycerol. This is again the properties of lipids, that they are hydrophobic, insoluble in water, soluble in ether, chloroform, benzene. So lipids contain carbon, hydrogen and oxygen, but the proportion of oxygen is much less, having only 2 oxygen atoms at one end of long molecule. Again the same properties, I am not going to discuss it. I will just only let you know the structure or the classification of the types of lipids. So we have simple lipids, complex lipids and derived lipids. So simple lipids are the esters of the fatty acids with alcohol. These can be fats as well as vaccines. So esters of the fatty acids with various alcohols. Now what is fat and what is oil? Fats is the ester of fatty acid with glycerol. Oils are the fats in the liquid state. Waxes, they are the esters of fatty acids with higher molecular weight, monohydric alcohol. Fats are the esters of fatty acid with glycerol. They are solid at room temperature. Some are liquid at room temperature, which are known as oils. Physical state depends upon the nature of the fatty acid. Fats are rich in saturated fatty acids. Oils are rich in unsaturated fatty acids. So you have to keep this thing in mind that fats are rich in saturated fatty acids and oils are rich in unsaturated fatty acids. So this is the example. This is a glycerol, CH2OH, CHOH, CH2OH. So along with this CH2 or along with this glycerol, three fatty acid chains are attached. So three fatty acid chains are attached. So we have according to the number of carbons, we have different types of fatty acids which are present. Like we have stearic acid. We have two types of fatty acids. We have saturated fatty acids in which we have no double bond in between the carbon, and we have unsaturated fatty acids in which we have the double bond in between the carbon atoms. So we have two types of fatty acids. The example of saturated is the stearic acid and the example of unsaturated is the oleic acid. So the double bond introduces a rigid band in the hydrogen tail. Fatty acid with one or several such bands cannot pack together as tightly as the saturated fatty acid. So we have saturated as well as we have unsaturated fatty acid. The unsaturated have the double bond. So these are the unsaturated fatty acids. Here we have the saturated fatty acid. Now these are the according to the number of carbon present, like 12 carbon, 14 carbon. We have different fatty acids. We have lauric acid, we have nitric acid, we have palmitic acid, stearic acid, arachidinic acid. The 16 carbon and 18 carbon fatty acid, they are quite important. That means palmitic acid and stearic acid. So they all are the saturated. So when we have a double bond in between, then instead of palmitic acid, they are known as palmitic oleic acid. Okay, so they have the double bond in between. So this is stearic acid. We have the double bond. They are known as the oleic acid. Then we have linoleic acid. Then we have alpha linonic acid. These are the foods which contains high fat, like butter, chocolate. They contain the high fat. Now we have storage lipids in which the fat is stored. They are known as the storage lipids. So we have the saturated fatty acid with no double bond, and we have the unsaturated fatty acid with the double bond. So the example is like glycerol. We are just attaching the three fatty acid side chains. So the examples of saturated is, we have to keep in mind the number of carbon atom. According to the number of carbon atom, we have acetic acid, butyric acid, caproic acid, capric acid, lauric acid, myristic acid, and then our stearic acid, palmitic acid. But when we have a double bond, then we have to say palmitoleic acid, oleic acid, linoleic acid, because of the double bond. So we have another term which is triglycerides, in which along with one glycerol, three carbon, three fatty acid side chains are attached. So the fats and oils are found in the animals and plants. They are called triglycerides. Triglycerides are the esters of fatty acids and glycerol. Okay. Triglycerides Triglycerides are esters of fatty acid and glycerol, simple lipids important as the storage form. Triglycerides are the esters of fatty acids and glycerol. Triglycerides are the simple Triglycerides are simple acids. They are important as a storage form of fat in the human body. So the fat which is stored in our body, it is stored in the form of triglycerides. So this is the glycerol. Types of glycerides, we have simple triglycerides, we have mixed triglycerides. So simply, they are the triesters made up from glycerol and three molecules of one kind of fatty acids. They are there. Mixed triglycerides, they are triesters with different fatty acids with different fatty acid components. Animal fats and vegetable oils are many different mixed glycerides. The example is butter fat contains at least 14 different carboxylic acids. So they are the mixed triglycerides. Getting my point? After it we have, so this is the glycerol CH2OH, CH2OH, CH2OH. So along with it, three fatty acid side chains are attached. These are some properties of fatty acids. The very first property is hydrogenation. So hydrogenation is the process of converting liquid oil to the solid fat by adding hydrogen to some of the double bond of the unsaturated carbon chain in the presence of nickel as a catalyst. So rancidity is what basically? Rancidity is when they develop the unpleasant odor and flavor on exposure to moist air at room temperature. So rancidity is caused by either hydrolysis of the ester bond or by oxidation of the double bond. So we have rancidity. Sonification of the fat is another term. So sonification is what basically? It is the hydrolysis of the fats and oils by boiling with sodium hydroxide. So by boiling with sodium hydroxide, we are getting the hydrolysis of fat and oil. These are again the fatty acids: acetic, propionic, butyric, valeric, lauric, palmitic, stearic. You have to keep the name of these fatty acids, remember. So we have some essential fatty acids also, which can't be synthesized in our body. So we have to take these fatty acids from the outside. Okay, that one are known as essential fatty acids. Now we have some lipids also. What are basically lipids? Complex lipids. We have some complex lipids also. It contains groups in addition to an alcohol and a fatty acid. We have phospholipids. Now what are basically phospholipids? So these contain, in addition to the fatty acids, they contain alcohol, phosphoric acid residue, and they are known as glycerophospholipids, sphingophospholipids as well. So the another term is glycolipids. Now what are glycolipids? Lipids containing a fatty acid, sphingosine and carbohydrates, they are known as glycolipids. Other complex lipids are sulfolipids. The example is lipoprotein. So these are the phospholipids. Like in the glycerol, we are having three fatty acid chains attached. Here what will happen? We have the phosphate group. So on the one glycerol carbon, we have the phosphate group attached along with one amino group. So these are the glycerophospholipids. After it we have phosphatidylcholines, which are also known as lecithins. Now what are these phosphatidylcholines? They are present in phospholipid of the membrane. Choline is the part which is present here. That is why they are known as phosphatidylcholine. The example is dipalmitoyl lecithin, which is very effective surface agent, more constituent of the surface tension. Then we have phosphatidyl inositol. So phosphatidyl inositol 4,5 bisphosphate constitutes our cell membrane upon stimulation by the suitable hormone. So this is the function of the phospholipids. Then we have sphingolipids. Now this is your homework. You have to tell me the difference between phosphatidylcholine, phosphatidylinositol or sphingophospholipid. So you have to make the diagram of all these fatty acids. Okay, saturated fatty acids, unsaturated fatty acids. You have to make the diagrams of all these fatty acids. Steroids we have already discussed detail in the hormonal part. So you have to tell me the difference in all these fatty acids. Otherwise the rest is same. So you have to tell me phospholipids, glycolipids. So we have two types of lipids. We have simple lipids as well as we have complex lipids. So you have to draw all these phospholipids, phosphoglycerides, phosphatidylcholine, sphingomyelin. So you have to draw the diagram of all these. Okay, phosphatidylcholine, sphingomyelin, phosphatidylcholine. You have to draw their diagrams only. Phosphatidylcholine, phosphatidylethanolamine, phosphatidylglycerol, phosphatidylserine. They all are the glycolipids basically. So you have to draw the diagram of all these. Then we have